The effect of constriction on cells, in particular at the level of the nucleus. Cell constriction is a phenomenon that has been reported in many processes, for example during migration of an immune cell in the extracellular matrix, as is represented here. These cells move around a lot in a crowded environment. To do so, they have to deform, which has an impact on the nucleus. It is the compartment of the cell containing the genetic material, called DNA. These deformations are studied in simplified systems, such as this microfluidic system where the cell encounters a bottleneck. At this moment, the nucleus is too big to move forward, and it is the action of an actomyosin network that will deform it to move forward. The nucleus will then be compressed, and this can induce ruptures in the envelope if the compression is too important. This induces an exchange of material between the nucleus and the cytoplasm, and can induce significant damage to the DNA. To prevent this, proteins will be recruited at the site of the rupture. First, BAF, barrier to autointegration factor, will bind to the DNA and recruit other proteins such as ESCART3 and new pieces of membrane to plug the hole. Altering the shape of the nucleus can have an impact without inducing damage to the cell, like in the mechanosensitive Shackling mechanism. When no force is exerted, a protein called YAP is found in both the nucleus and the cytoplasm in the same concentration. It can pass from one compartment to the other through the nuclear pores, which are covered with FJ repeats, in purple here. When force is applied, the nuclear envelope stretches and the nuclear pores deform as they open to the exterior. The cytoplasmic YAP proteins interact with the FG repeats of the nuclear pore, inducing YAP unfolding and facilitating their entry in the nucleus. Because it happens only for the cytoplasmic YAP, it will induce YAP nuclear accumulation and modify gene expression in the nucleus, modifying the cell's activity to adapt to the constriction. In a mechanism called the nuclear ruler, nucleus compression can induce release of arachidonic acid and calcium from the nucleus. Both upregulate actomyosin contractility at the cell membrane. This could be used to sense cell height or to increase migration speed by forcing on the surroundings due to the enhanced cortical contractility.